Great, thanks so much, Rana. I really appreciate the opportunity to talk at this first virtual PHUSE seminar. Before I get started, I wanted to, so I try to introduce some interactivity, given that we're no longer meeting in person. So if you're able to just visit this website that's posted here, I'll have a couple questions along the way that'll hopefully foster some communication between all of us throughout the session. And at the end, of course, we'll have some time for questions as well, but I wanted to introduce some opportunities throughout the presentation. So as Rana mentioned, I am a quantitative scientist at Flatiron Health, and Flatiron Health is a healthcare technology and services company that is focused on accelerating cancer research and improving patient care. So briefly, I work on a team that manages a clinical genomic database, but today, for the most part, I'll be talking more generally about the growing role of electronic health records in pharmaceutical research with a focus on cancer treatment, as that's where I've been working the most, but I think most of these thoughts apply fairly broadly throughout the use of real-world evidence in pharmaceutical research. So the points really go down to these three things. So kind of introducing EHR data generally, and then talking specifically about the data model at Flatiron and how we translate that into real-world evidence that can be used for insights. Then I wanted to delve into a few example use cases of real-world data in drug development and patient access as well. And then for the most part, I know that many people on the line are data scientists, is to glean some key analytic considerations that I think are important when you are gathering insight from EHR-based real-world data. So that's the bulk of the presentation, and then at the end, I definitely want to answer any questions or have any discussions if anyone is interested. So Flatiron really has two sides to its business. First is a provider side that implements a cloud-based EHR for specialty cancer clinics, so mainly smaller community sites around the country. And we also have an academics integration platform that allows larger academic medical centers to share data with us and allow us to share data back with them as well for both research and their own quality improvement. So that's the first side, it's the provider side. The second side, which is where I and my colleagues work, is the research side. And so we work with life science partners, with regulators, with health technology assessment organizations to use this data that we've established. So this slide shows the pathway from getting data to evidence, starting with the EHR. As I'm sure a lot of you know, EHR data consists of both structured and unstructured data elements. Within this unstructured data, we have things like free text and scan documents, so things like pathology reports, physician notes, radiology reports, and scans that are actually copied into the EHR. Structured data, as you might guess, is much easier to process. That is really just taking them and coalescing them into and normalizing them into analyzable formats. The unstructured data processing part is really the more challenging part that I'll talk about just briefly in a second. Both of these elements are then put into a research database that we use, and sometimes we'll conduct data linkages. So two examples of these are with external mortality data or with genomics data. And then we also provide analytic platforms on top, as well as support for conducting analyses down the road. So I mentioned that key data elements in oncology are largely in the unstructured data. So so elements such as staging, the diagnosis date for patients, biomarker statuses, clinician reporting on response or treatment progressions. These are really only revealed in unstructured data. And so here's an example of a PD-L1 IHC report, which has a lot of these variables, but it's a scanned document. It's on its own, it would be otherwise very difficult. You might have to conduct an ad hoc chart review or some other time-consuming process. So these are some of the variables that we would potentially put into a table that would allow researchers to use for analysis. And again, yeah, most of these clinical data points are captured in EHR. So similar to a trial, we can conduct research with them if we're able to glean these unstructured data elements and unite them with structured data elements. So for example, here's a patient who might be diagnosed with early stage cancer, and then we have notations about surgery that patients might have. We then might also have a clinic note that they developed metastatic disease, that they were tested for certain standard of care biomarkers. They started their first-line therapy, and there's progression They were tested for an EGFR resistance, and then we have additional therapies they might have taken, and then unfortunately, a death outcome might be noted in the EHR. So these are all things that we capture in our data, and I think most EHR-based real data sources will capture, but I think this is an example of just the flow of how we can capture the entire clinical journey for a patient. Briefly, I wanted to mention the product I work on. This is one of our products that we work on that uses external data as well. So we have our Flatiron EHR data, which we unite with genomics data from a company called Foundation Medicine and now we have over 50,000 patients where we're able to have this enhanced clinical real-world data that's united with comprehensive genomic data from Foundation Medicine. So it allows us to segment patients by the cancer types they have or certain biomarkers or diseases within disease models or within a whole pan-tumor space. So those are the last of the slides talking about the Flatiron data specifically. Now I wanted to kind of go into EHR data more generally. And just to prime you for this, if you're able to go to this website now, the next page will have a short kind of survey for you to see how you might have used this data before. But I wanted to highlight these 5 major areas where we've viewed EHR data as being useful and used in the past across the biopharma space. First of all, in trial design, where you have, we know that RWD can provide a really strong complement to trials, especially if historic trial data is not available or is not contemporaneous with the trial that's being conducted. So there's different ways to assess control arm assumptions or look at treatment patterns to help choose a standard of care for a control arm. Also helping to guide how regimens are selected, looking at similar compounds in EHRs for how dosing is conducted. Also for regulatory submissions, it allows you to characterize certain populations, unmet need, the natural history of patients without, say, a certain treatment that might be under investigation. And with HTAs, we know that EHRs can capture longitudinal outcomes, sometimes better than trials, because trials may only be conducted on a small population or for a short period of time. So we know that EHR really can help support looking at some of the outcomes that are important to payers. So overall survival, some other patient-reported outcomes potentially, which can support reimbursement and pricing discussions. The last one, which I think fits into a lot of these other ones, is scientific publications more generally. So understanding the characteristics and outcomes of patient populations, assessing and validating real-world endpoints, and also conducting methods research. We know that we can complement simulations, and a lot of the studies that we've published, some of the trial replication work can be really interesting to see if you can set up a control arm or even a treatment arm for patients using real-world data to see if it is aligned with what was conducted in a trial. So these are all different considerations that are useful down the road. So, all right, so the next slide I'm going to leave up for a second, and it's going to do an interactive poll here. So I would love to see just generally who in the audience, and we have a couple hundred people here, what settings you have used EHR-based RWE, just to get a sense of where in the spectrum you're coming from here. So we'll give you a second to kind of go on and vote if possible, and then we can see if we have any results here. Okay, great. So we have, this is changing rapidly. So a lot of people seem to have focused on presentations. Seems like it's growing as people have not used EHR data yet. So we'll give it a few more seconds here to balance out. I think you can vote about 5 times. So if you have multiple use cases, feel free to change it. But give it another second. Okay, great. So let's lock it now. Give maybe another second or two. So it looks like it's pretty balanced across all these spectrums. So it's interesting to see that there's work in complementing trials, but also potentially supplementing trial data as well. All right. Great. So then I wanted to jump into through examples of how RWD at Flatiron has been used in a couple settings. So the first one I want to talk through is label expansion. So we know, so this is particularly useful where there's potentially off-label use of drugs, but not necessarily yet an FDA approval. So just to get background on this study, palbociclib was originally approved in 2015 for women with advanced breast cancer. And we know that breast cancer is very common, but male breast cancer patients are generally just not included in trials because they're a very different type of patient, and conducting trials on males alone would be very difficult to do. So we engaged Pfizer to explore these baseline characteristics and outcomes for a group of male metastatic breast cancer patients. And Pfizer included patient-level data to support the efficacy, looking at response and other variables, as well as the safety. So looking at really detailed adverse events for palbociclib as part of their FDA submission. So FDA last year approved a label expansion for palbociclib based in this population, and Pfizer presented the results at ASCO 2019. So this is one of the cases that shows what aspects are important for regulatory agencies to evaluate in label expansion. So looking at adverse events and any potential differences in outcomes based on the new population that's under study. The second, this is the last example I wanted to show, was how control arms can be used to secure reimbursement. And in this case, with EMA and other health technology assessment organizations. So following a single arm study, this is for alectinib in non-small cell lung cancer in the crizotinib failure setting. Roche submitted for EMA approval for the drug in advance of their pivotal study. So to secure reimbursement, they needed to demonstrate the superiority of alectinib to ceritinib, which was currently the standard, which was the standard of care at the time. So using Flatiron real-world data, Roche was able to generate an external control arm of patients who received the then standard of care, and then enabled a comparison to the alectinib trial data. So then they weighted this cohort to reflect the population that was in the trial, to adjust for these differences in baseline characteristics and allow them to show median survival and the survival outcomes more generally in comparison to the trial arm. So the results helped to expand access in over 20 countries over a year earlier than anticipated. So the next slide will show you the differences that we saw in the groups, the alectinib from the trial arm compared to the ceritinib arm from the real-world data. So as an interesting follow-up from that, the external control arm from the Flatiron real-world data, which you see here is this lighter green curve, was then compared with the ceritinib arm of the trial that was published later. And we can see that the curves
Jeremy, can you hold on for a second?
But I just want, yes.
Yeah, so just a minute ago, we lost your audio. I think you're back now. Would you mind just repeating the last 60 seconds, maybe?
Sure, yeah, I'll just do this slide again to talk about how when Roche had conducted this analysis of the ceritinib arm in the real-world data, there was an updated report from a trial, this ASCEND-2 trial, which was a trial arm for the ceritinib in the setting, which showed a very similar OS, median OS, to what was seen in the real-world data. So it shows the value of both real-world data, but also adjusting the cohort to make sure that you're reflecting the patients that might be in the trial. So hopefully that summarized this slide. I'm happy to go back to it later if there's any questions. Great, so as I mentioned, I'd love to get some feedback on this slide as well, but I'll lay out this case study, which which is a case study that we have dealt with frequently in the clinical genomic database that we work with. When we're talking with clients, there's a lot of interest in questions like this. So I'm curious how this resonates with the PHUSE audience. So let's say that your company has developed a new drug that will treat patients with a certain cancer who show alterations with biomarker Y, we'll say, which is identified. It's not a standard of care biomarker, but it could be potentially identified using next generation sequencing. So you had conducted a single arm trial which was only with patients with this biomarker, but you're interested potentially for regulators or HTA submissions down the road to understand the unmet need of this population. You don't know necessarily how this biomarker Y population will have the outcomes of this population normally in the absence of the drug. So you want to benchmark your trial results to this and just understand the unmet need generally. So historical trial data is not really available or it's outdated and doesn't really represent the standards of care that we see now for the population. So you turn to real world data sources. So my question for you is how would you evaluate whether RWD is appropriate for this population? So I'll give you a few seconds and go to the next slide and just think about like what are some questions you might ask to determine an appropriate role for EHR based real world evidence. So are there any things that you can think of that you would want to focus on in thinking about what's important? I'll give a few seconds and we can come back to this if people want to think about it a bit more. But let me just prompt some ideas as we go forward here, and then we can see if we want to talk about it later. So we've generally identified we've lessons learned and internal guidance is from discussions with regulators and with clients about how we might want to get at these considerations. These are generally undergirded by pre-specification generally. So I think all of these bullets will fall into, will be affected by pre-specification. It's just a good scientific practice generally, and we know it's important in academic settings, but it's crucial in regulatory settings. Clinical trials need to have it, and real-world data analyses are generally things we want to approach in the same way. And it underlies these four other areas, which I'll get into in more detail. So the four areas we've identified are really relevance of the data and making sure that your data is high quality, that you're appropriately selecting a cohort, that your real-world endpoints are suitable for the question you're asking, as well as the analytic methods you're using are suitable. So Rebecca mentioned this in the first presentation of this idea of fit for purpose data. So we're using this framework that was developed by the Duke Margolis Center for Health Policy, which does a lot of work in partnership with FDA, as well as collaboration with a number of groups, including Flatiron, to come up with standardized ways to measure data quality and to measure fitness for use. So this, as you can see, these fit into some of the other buckets we're talking about as well, but they center around the availability of data, making sure it's representative, that you have an adequate cohort size to conduct an analysis successfully, as well as adequate follow-up time. Then the accuracy, completeness, the traceability, the provenance of the data, as well as transparency in how your data was, your analysis was coded, how you arrived at the cohort, and so forth. So within this, you can get at fit for purpose data, but there really is no one-size-fits-all solution for these types of questions. So it really just starts with a well-defined research question, and then you can evaluate the critical data components as you move along in the context of that research question. So next, I wanted to focus on a brief example of data quality, which is part of that framework. One is the completeness. This is a potential. It's a potential concern with EHR because we know that EHR data is designed to support clinical care and workflows, and data can be missing from a research perspective, but complete from a care perspective for a variety of reasons. So understanding data completeness is critical to assessing the fitness for use. So we know that EHRs might just not have elements that are important for your study. The data might be available in documents that are buried in the EHR, but not in structured data that could be easily analyzed. So that's one of the things we talked about earlier in how Flatiron is able to translate some of that data that is in unstructured fields into analyzable workflows. There also could be variability across sites. There's no standard protocol in many cases for how you describe endpoints or exposures of interest. So those are things that need to be accounted for in your analysis. Just a general way to think about missingness, there's this looking at the extent of the missingness you're seeing, so the proportion of patients that have a value that you can use for an analysis. But I think another thing that's just as crucial is to assess the criticality of the variables. So this means how essential are they for your analysis, and how much would it affect your analysis if it was missing or potentially erroneous value. So cohort inclusion-exclusion criteria that might match trials, such as correct ECOG scores, are essential because that defines your cohort and can potentially lead to selection biases if you just exclude patients with missing values, for example. You need to be really conscientious in how you do that. The second thing is, if you have an exposure definition, if you're looking at two groups, you want to make sure that the actual exposure that you're looking at is captured correctly and completely. And then outcomes or endpoints are also going to be very important. So this could include index dates used for starting an overall survival analysis, as well as the endpoints associated with those as well. So now I wanted to talk a little bit about cohort selection, which I think fits in to some of the points we've talked about earlier. We need completeness in data quality to select the right cohort, but once you assess the data, you still want to make sure that you are selecting the cohort appropriately. So you want to make sure that you identified and captured the clinically meaningful variables. We know that aligning on an eligibility criteria that might match a trial can result in significant attrition in the cohort size, as well as a change in the type of population. So you want to understand, are you trying to get a cohort that matches what you see in a trial, or do you want to look at a population that might allow you to understand the effect of a drug on a broader population? So kind of leaning into that estimand framework of understanding what is the treatment effect you're looking at. Also, it's really important to incorporate methodologies that will address missingness as well as potential biases. And I'm going to talk about some of those methodologies in a second. And going back to the framework as well, the Duke-Margolis framework, having a transparent and traceable selection process that is documented well is essential for regulators and HTA bodies in the cohort level. We know cohort selection is a crucial component for a lot of the regulators and HTAs that we've worked with. Some of the other additional considerations that we think have really been important is representatives of sites. So we know that if a trial is limited to a few sites, single institutions can have potentially a large influence in outcomes, potentially leading to a bias towards a false positive finding and overhyping of results in some cases. So there are strengths when you're increasing representativeness of patients that, and we avoid those outlier sites that might change your interpretation. Concurrency of treatment. So we know that standards of care change over time. So we know if we choose patients who might be in this bucket, they're going to have different standard of care than patients that might be in the concurrent window for the trial recruitment arm. So we know that comparing patients within a similar time frame of the standard of care treatment will potentially yield a less biased comparison. The last thing is timing of cohort eligibility. We know that if we're defining a biomarker or defined arm or some other EHR-defined arm, we know results might come at different points in their treatment, and including patients that might be tested for, say, a biomarker at baseline or several years after their initial diagnosis might yield a very different patient population. So characterizing the quality performance of these endpoints is essential. So are the measurements reliable? Can they be benchmarked? So at Flatiron, we know we've looked at mortality as a variable, and it seems like a very straightforward variable, but we found that just within the EHR itself, using structured date of death information, it did not capture the totality of mortality events. So we've, in this paper that was published by Curtis et al., we identified this 4-part composite index of mortality variables. So using unstructured documents, commercial death data sets, as well as Social Security death information, we can get at a very sensitive and specific death measure. So even variables like mortality that might seem fairly straightforward to use can definitely use validation. Another brief example I wanted to show is with response. So we know that RECIST is a key criteria in many cancer trials, especially phase 2 trials or early research that aren't focusing on OS or PF, progression-free survival outcomes. But we know that RECIST is really tightly defined, both in terms of the protocols used for analyzing the scans and the timing of the visits. And we know that in real-world data, we don't have a protocol for issuing these scans, so it's really based on patient need or clinician preference. There's no interpatient variability as well. The last thing I wanted to focus on is just some of the analytic methods that we are thinking about at Flatiron and working with our clients and with regulators and academic institutions to think through how we handle them effectively. So first is, we know that small cohorts can arise when we are limiting to a very small biomarker-defined cohort. So thinking about ways to quantify the uncertainty in these cohorts, as well as constructing patient narratives or other ways to effectively to view these cohorts. Some of the things, this is more general, is just dealing with selection bias and also confounding of variables. So finding ways to describe these patients better and use balancing methods to when you're comparing to, say, a trial or another data source. The last thing I want to focus on is it's sometimes not as fully understood, I think, by a lot of consumers of real-world data, is if you are combining data across different sources, this question of delayed entry or left truncation comes up, where if you are relying on patients surviving long enough to get into all the data sources that you're using, it can lead to overestimated survival time. So we're currently investigating methods to adjust survival estimates to make sure that you are not producing over-biased risk samples, and or thinking about other ways to structure your cohort. So you're removing that bias as well. So my overall conclusions are really, I think the paradigm is evolving rapidly for drug development. Standards of care are changing. Retrospective, perspective, and observational interventional research lines are being blurred. We know that Flatiron is launching some prospective studies on our data, and we're heavily combining observational and interventional data sources in our analyses. And we know EHR data can provide research and regulatory grade evidence and complement evidence from clinical trials. So yeah, those are all of the points I wanted to make. So thank you very much for the opportunity.